Good afternoon to one and all. Myself Jyothi, working as Assistant Professor in Pharmaceutical Engineering Department. Today I am going to discuss a topic on Pharmaceutical Analysis Subject Introduction. The main course objectives of this pharmaceutical analysis subject to provide knowledge on the qualitative and quantitative detections for various pharmaceutical samples. Say, for example, if a product is there, means we need to find out the quality and the quantity. Before going to the formulation of particular formulations, we need to find out the qualitative and quantity checking of that particular product. For the preparation of any pharmaceutical ingredient, pharmaceutical formulations, we need to check the quality and quantity of that particular formulation. For that purpose, we need to study this pharmaceutical analysis subject. And second objective is that to impart the basics of working and applications of UV visible spectroscopy. In a first point, we find out this qualitative and quantitative detections of particular API compound. We discussed how we will detect the quality and the quantity of that particular API ingredient. For that, we required one equipment. That equipment is a UV visible spectroscopy. By using this UV visible spectroscopy only, we can find out the quality and the quantity of that particular compound. Next, third objective of this subject is that to expose the students to identification of the functional group present in the sample using IR spectroscopy. Here, in a pharmaceutical analysis subject, we can find out that functional group of that particular compound also. For the quality checking and the quantity checking of this particular API, we need to find out the functional groups, particularly that functional groups are identified by the IR spectroscopy equipment. This is also one of that analysis. Next, to understand the principles involved in the NMR and the mass spectroscopy. This is the one more technique used to find out the quality and the quantity of that particular compound. NMR and the spectroscopy. NMR spectroscopy and the mass spectroscopy. These are also qualitative techniques to identify the unknown sample containing the concentration and the functional groups present in it. And next, to introduce the different chromatographic techniques. These chromatographic techniques are used to separate the compounds. If the formulation is having any unknown samples, means we need to separate it, right? By using a chromatographic techniques, we can separate the samples and we can analyze that functional groups also by using these chromatographic techniques. These are the main objectives of this particular pharmaceutical analysis subject. Coming to this introduction of this pharmaceutical analysis, as the objectives we discussed, pharmaceutical analysis means is a branch of chemistry. It involves a series of the process. In a series of process, we need to identify and we need to quantify and we need to check the quality of this particular compound. Right? The analysis means identification of this functional group, determination of the particular functional group, and the quantification and the purification of a substance, and separation of the compounds of a solution in a mixer, or determination of the structure of this chemical compounds and the pharmaceutical agents. All these analyses are done in the pharmaceutical analysis subject. In a identification process, in a determination process, in the quantification process, and the purification process. By using all these analytical techniques, we are going to perform the pharmaceutical analysis for a particular formulation. And another definition for this pharmaceutical analysis is also defined as that applications of this analytical tool. By using analytical techniques, by using analytical tools, we are identifying the unknown quantity and the quality of this particular unknown sample. See, here also techniques and the procedure used to determine the purity, safety, and the quality of this drug and the chemicals. Studying about all these parameters comes under the pharmaceutical analysis subject. Coming to this next one is the scope of this analysis, where we can apply this pharmaceutical analysis subject. That is the scope. We can apply this pharmaceutical analysis subject in a pharmaceutical industries, in a food industry, in a cosmetic industry, in disease diagnosis cases. We can use this pharmaceutical analytical techniques, right? In a pharmaceutical industry, in how these pharmaceutical analysis techniques are applying in a pharmaceutical industry means for the identification of excipients and for that preparation of that different dosage forms. For that purpose, they will use this analysis techniques in a pharmaceutical industry, and for this identification of this quality and the required quantity for the preparation of formulation. For the preparation of formulation. All these things are done by the pharmaceutical analysis, pharmaceutical industries, this subject. In food industry, in food industry, this pharmaceutical analysis subject is useful means for the identification of preservatives levels. For the food preservation purpose, in food industries, they will use these preservatives. So these preservatives, without these preservatives, this food will not be stable. It is going to be spoiled. At what level we need to add these preservatives to this particular compound? If any food containing these preservatives, which type of preservative it is having? For that identification, we need to use these analytical techniques. Next, in a cosmetic industry. In cosmetic industry also, lipsticks, especially lipsticks, it is having a color agent. It is also one of the chemical. At what level, at what quantity we need to add this coloring agent to this particular lipsticks? That quantity we can identify by the analysis techniques. And perfumes. Adding of perfumes to this cosmetic product, the quantity, the quality of this perfumes we can identify and we can check by this analysis subject. In disease diagnosis cases, HIV, this human immunovirus, this HIV is identified by the ELISA techniques. This ELISA technique, it comes under the analysis subject. So like these are the scope points, and this is the scope for a pharmaceutical analysis subject in a food industry, cosmetic industry, pharmaceutical industry, and a disease diagnosis conditions. Coming to this next one, up to now we discussed about the pharmaceutical analysis subject for the identification of the quality and the quantity we discussed. Right? The different techniques to identify the analysis is that one is that quality and the next one is that quantity. There are main two types of chemical analysis. That one is that qualitative analysis and the quantitative analysis. In a qualitative analysis means identification of the compound, which functional group is present in that, which type of excipients are there. This quality, this identification is done by the qualitative analysis. In a quantitative analysis, it's an estimation. How much concentration is there? Always it is denoted in a numerical format. Always it is identified in a numerical format. This is quantitative analysis. See, in a qualitative analysis, these tests are performed to indicate whether the substance or compound is present in the sample or not. Functional group, required functional group is there in a particular API or not. In a particular API, required functional groups are there or not. That is checked by the qualitative analysis method. In a quantitative analysis, techniques are mainly used to quantify the compound or substance in a sample. In a particular API, how much concentration of pure compound it is, we can find out in a quantitative analytical method. There are so many techniques to identify the unknown samples in a pharmaceutical industry, food industry, cosmetic industry. These are the analytical techniques. Non-instrumental analytical techniques and instrumental analytical techniques. Non-instrumental analytical techniques means without using any equipment, without using any instrument, identifying the quality and the quantity of that particular compound. Like in that, various chemical methods are there. And in biological methods, in vitro methods, animal studies, microbiological assays, by using these methods, we can find out the particular functional group and the concentration. Our pharmaceutical analysis subject, it denotes what? Identification of the quality and the quantity, right? This identification of the quality and the quantity in a biological methods means in vitro, animal studies, and the microbiological assays. By using these methods also we can study. Instrumental analysis. By using some equipments, by using some instruments, we finding out that quality and the quantity of that particular sample. Instrumental analysis techniques are volumetric analysis, electrochemical analysis, spectroscopic analysis, chromatographic analysis, hyphenated techniques, and the other methods. All these are the analyzing methods. By using these analyzing methods, we can find out that unknown sample containing functional group and the concentration. Coming to this volumetric analysis, in volumetric analysis method, as the name suggests that titration volume is used. So it is a quantitative method. Titration volume it is used means how much amount of that volume is required to neutralize the in a volumetric analysis, it is mainly called as the titration methods. In a titration methods, here one burette will be there and conical flask will be there. In a burette, acid or base will be present. In a conical flask also, acid or base will be present. By using neutralization as a principle, we can find out that concentration of that particular sample. That is a titration volume. So it's a quantitative method. How much quantity is consumed for the neutralization? How much quantity of base is required to neutralize the acid for a neutralization? Here concentration, quantity. That's why it's a volumetric analysis is the quantitative method. Volumetric analysis are again classified into acid-base titrations. In acid-base titrations, aqueous titration, non-aqueous titration, second redox titrations, complexometric titrations, precipitation titrations, all these are concerned the volumetric analysis. This is that one of the method to identify, to quantify the particular unknown sample present in a formulation. Next one is that electrochemical analysis. Here in electrochemical analysis method, analytical techniques that are used to measurement of the potential, charge, current to determine an analyte concentration. In a previous slide, in a volumetric analysis, with the help of volume, we find out that unknown concentration. There neutralization as a principle. In case of this electrochemical analysis method, here we are measuring the potential, charge and the current to determine the analytical concentration by using potentiometric titration, conductometric titration, amperometric titration, polarography equipment. By using all these equipments, we can find out the potential and the charge, the positive charge, negative charge present in that particular formulation or having particular functional group. That we can find out in an electrochemical analysis method. Next one is that spectroscopic analysis. In spectral analysis, spectroscopy can be involved in any interaction between the light and the matter. With the help of light and the matter, using as a absorption, emission and the scattering as the principles, we can find out the concentration of the unknown sample. Concentration of the unknown sample. Here, the light and the matter. Light means it is a source and the matter means it's an unknown sample. How much amount of that light is absorbed by the unknown sample, that is that absorption, and how much amount of that light is emitting from that particular unknown sample, that is that emission, and how much amount of that scattering is possible from that unknown sample. By using these three parameters, by using these three principles, this unknown concentration of the sample will be identified in the spectroscopic method. The four techniques commonly used to help the structural analysis is visible spectroscopy, UV visible spectroscopy, and infrared spectroscopy, nuclear magnetic resonance spectroscopy, and mass spectroscopy. By these four methods are involved in a absorption, emission, and the scattering principles. By using these three parameters, it is giving the unknown sample concentration. That is the spectroscopic analysis. Next one is the chromatographic analysis. In chromatographic analysis, as we discussed in the introduction slide, this pharmaceutical analysis subject is used to qualify and the quantity, used to identify the unknown sample quantity and the quality, and at the same time we can separate that unknown functional groups like this we discussed. For the separation of this particular samples, we need to use this chromatographic analysis techniques. Earlier we discussed spectroscopic, electromagnetic, and volumetric analysis like this we discussed in a previous slide. There we find out that only functional groups and that quantity and the concentrations we find out, but the separation of this two different compounds in a solid or mixer, in any other form, we can separate the compounds by using this chromatographic analysis method only. Right? This chromatography, it's an analytical technique commonly used for the separation of a mixture of chemical substances into individual components. This chromatographic technique, it works on the absorption principle and the partition principle. In absorption principle, competition between the solid and the gas, solid and the liquid. By using column chromatography, we can separate that solid to the liquid. And thin layer chromatography, by using this thin layer chromatographic technique, we can separate the solid and the liquid. Like that, in a partition chromatography, liquid and the gas, we can separate in the liquid form HPLC, liquid. If unknown sample is present in a liquid, means by using HPLC technique, we can separate the two components. In a column chromatography, we can separate that liquid in a liquid sample, and the paper chromatography, liquid in a liquid, and thin layer chromatography. By using these different methods, we can separate the components and we can find out that functional groups. We can find out the functional group present in a synthesis phase. Next one is that hyphenated analysis techniques. These hyphenated techniques are those analytical techniques in which a chromatographic technique and the spectroscopic techniques are used to analyze that various biological samples, chemical samples, and the toxicological samples. See, it's a mixed technique. It is by using chromatographic technique and the spectroscopic techniques. By coupling of two different analytical techniques, we are finding out the analyte in a different compounds. GCMS, gas chromatography, and the mass chromatography. Here we are coupling two different analytical methods, and LCMS and LCNMR and GC and NMR, nuclear magnetic resonance equipment, and the gas chromatography, liquid chromatography, and the mass spectroscopy, and ICP and OES, inductively coupled plasma and optical emission spectroscopy by using an ICP-AAS, atomic absorption spectroscopy. These are two different methods. By using two different methods, we are identifying the analyte present in the biological and the chemical and the toxicological substances. This is about pharmaceutical analysis subject introduction.
Like, share and subscribe. Hit the bell icon for more updates.